Okay, well, thanks for joining us, everyone. Welcome to this afternoon's Wheatland seminar. Most of you know, my name is Tony Guay. I'm a remote sensing specialist with the Wheatland Lab in the School of Forest Resources. And we're very excited to have Dr. Anna Schweiger with us this afternoon. She's a postdoctoral researcher in plant and vegetation spectroscopy with the University of Zurich and also affiliated with the University of Montreal. So with that, Anna, I'll turn it over to you and we'll go from there.
Great. Hello, everybody. Thank you so much for the invite, Tony. It's really a pleasure to be here with you guys today, virtually at least. So let me get started here.
No, we're seeing our presenter notes too.
Yeah, is that better?
Yep.
That looks good. Yep.
Okay, wonderful. So yeah, this afternoon I'm going to talk to you about spectroscopy of plants and what light spectra can tell us about functional, taxonomic, and phylogenetic dimensions of plant biodiversity. I want to start with thanking the great bunch of people I've worked with over the last couple of years. Etienne Laliberté from the University of Montreal, Janine Cavender-Bears from University of Minnesota, and the PIs from the Dimensions of Biodiversity project, Sarah Hobby, Phil Townsend, and John Gammon. This project was also led by Janine. And of course, Dudu and other collaborators that worked with me through the time. So, and many helpers in the field, of course, too. So, spectroscopy, what is spectroscopy? Spectroscopy is defined as the study of the interaction between electromagnetic energy and matter. The history of modern spectroscopy goes back to the earliest 19th century. In 1817, Joseph von Fraunhofer discovered dark lines in the solar spectrum, and he also measured the wavelengths that are associated with those lines. Later, Kirchhoff and Bunsen noticed that these wavelengths coincide with characteristic emission lines of heated elements. And they rightfully concluded that the absorption features in the sun were caused by the chemical elements of the sun's atmosphere. Since then, astronomers have been using absorption features of elements to study the chemical composition of stars, planets, and their atmospheres. And today, spectroscopy is important for the search of habitable planets and signs of life beyond Earth. I'm probably not the only one in this room who is fascinated by space science. And I'm also all for exploring all options to survive the Anthropocene. However, at the same time, I'm really a big fan of planet Earth. I think we have an extraordinary planet, and that it is, you know, a scientist our joint responsibility to protect and restore the ecosystems using all the science and all the tools we have. And we are lucky in that regard. After many years of joint efforts by the remote sensing and biodiversity communities, space agencies are now investing in a fleet of sensors for Earth observation. They include instruments like JEDI, high-resolution LiDAR, EcoStress, that's going to be measuring evapotranspiration, OCO3, measuring sun-induced fluorescence, as well as a set of 3 imaging spectrometers, and I'm particularly excited about those. NASA is planning to launch a free flyer, SBG, approximately, or you know, the envisioned time frame is the middle of this decade, and NASA's SBG will be covering the Earth at a time interval of about every 16 days and deliver spectral information, which is pretty exciting. Why do we need these huge investments in Earth observation? Well, one reason is that when it comes to predicting how biodiversity is going to respond to global change, we are actually not doing such a great job. The problem starts with the fact that we don't even really know where most plant species occur. And you've probably all seen graphics like this that illustrate that many areas of the globe, in particular in the global south, and also here in Central and Northern Asia, are vastly understudied. What adds to the challenge is that plant species are actually changing their distributions right now. And it is very hard for us to keep up with the pace of these changes with traditional field sampling. Very hard to, I would say impossible. So what we need, what we dream of, and by we, I'm talking about, the Earth observation biodiversity community, is a global biodiversity exploration and monitoring system. Ideally, it would integrate field and remote sensing data collected across different scales and provide spatially continuous, publicly available and accessible, high spatially and temporally resolved information on biodiversity, as well as ecosystem function and health. With remote sensing, we can observe plants from above. So let me ask you, why do we focus on plants to study biodiversity and ecosystem function? Well, first of all, plants don't move very much and they can live pretty long. So over their lifetime, they integrate environmental factors within a certain space and over time, and capture a range of synergistic as well as antagonistic effects of microclimate, soil, as well as disturbances and species interactions. This integration of environmental factors consists really of a constant trade-off of energy investment that plants do, energy investment in growth, seed production, nutrient storage, and the development of protective structures. And we can measure these investments, for instance, when we look at plant functional traits. Trait-based ecology really provides us with a framework for analyzing different life history and resource use strategies, and also competitive strategies and how they influence ecosystem function. Vegetation spectroscopy promises the detection of plant traits and trait syndromes, which are collections of traits, through measuring the light reflected from plants. And it promises to do this across scales of measurements, starting at the leaf level. And, since spectrometers are, in essence, very specialized cameras, they can be employed or clipped with a leaf clip directly to the leaf level, but also mounted on different platforms, such as drones, airplanes, and ultimately satellites. How we can harness the information of provided by spectroscopy for biodiversity science is going to be the content of my talk. And I'm going to spend the first couple of minutes to give you a short spectroscopy primer and talk about the prediction of plant traits from spectra. Then I'll be moving on to the topic of linking plant spectra to the tree of life. And in the end, I'll be talking a bit about this relatively novel idea of using spectra as integrated measures of plant phenotypes. Okay, let's start with the predicting plant traits from spectra. The sun emits electromagnetic radiation predominantly between 300 and 3000 nanometers. About 5%, I don't know if I have a pointer, maybe not. No, sorry, I don't have a...
You do, yep.
I do?
Yeah.
Oh, great. So about 5% of the radiation fall here in the UV portion of the spectrum, 40% in the visible, and 50% in the near infrared. And this is exactly the range where we generally do our spectral measurements. When sunlight interacts with any tissue, including plant leaves, two things can happen. Part of the radiation gets absorbed and part of the radiation gets scattered, either in backscattered, which is also called reflectance, or scattered in forward direction, which we call transmittance. Absorption is a wavelength-dependent process and is caused by molecular movements and the stretching and bending of molecular bonds. And this all depends on the chemical composition of the elements we're talking about. Scattering, on the other hand, happens when light gets deflected from a straight trajectory as it crosses the boundary layers of different materials, such as in plant leaves. We can think of light crossing the layers between the cells and plant cells and intercellular spaces. So this means that reflectance at the leaf level is influenced by leaf chemistry, anatomy, and morphology. And when we look at the canopy level and measure spectra from a distance, spectral reflectance is also influenced by the architecture of the canopy, because scattering also occurs within the canopy. This is a typical reflectance spectrum of the leaf, and you can see that plants are very efficient at absorbing reflectance in the visible part of the spectrum, and they of course power photosynthesis with that energy. And yeah, here you can see the absorption features of photosynthetic pigments. They absorb less here in the green region. This is why we observe here this reflectance bump, and this is why plants look green to us. Other characteristics are high reflectance in the near infrared, caused by leaf structure mostly, and we also see characteristic absorption bands of water. For instance, here on the left, I'm showing, the change of spectral reflectance as a leaf dries out. So on the top is a desiccated leaf, and as leaves dry, these absorption features here, they get more and more shallow. So this allows us, for instance, to detect drought stress from spectral measurements. And then, of course, there are many wavelength regions that are influenced by, all different kinds of molecules. And I'm just pointing out a couple here, a couple of regions that are known to be influenced by lignin content of phenolics, proteins, and yeah, here another lignin feature. Imaging spectroscopy data, so spectral data collected from a distance, is typically illustrated in this reflectance cube. So here we can imagine this as, an X and Y coordinate in space, in landscape space, if you will, and the Z coordinate is the spectral dimension. So each of these pixels in an image has, associated with it a reflectance spectrum, and it looks slightly different to a leaf spectrum because it's, in addition to, leaf chemistry, also influenced by canopy structure or soil background effects, some rest of, some remainders of atmospheric effects, depending on how the data is corrected, and so forth. I want to quickly illustrate a typical workflow of predicting chemical and structural traits from spectra, because I don't know how familiar you guys are with this. Let me walk you through this. Imagine we have our study area here. This is, for simplicity, let's think of it as a grassland, because I worked a lot in grasslands. That's always easier for me. So we are interested in, the vegetation composition in this research area. What we would typically do is establish a couple of research plots. These are the red squares here, and then cut the vegetation in these plots, for instance, and determine the trait of interest. Let's say it's nitrogen content in the lab. Then we would match the nitrogen values with pixel level reflectance, or the reflectance values from the pixels within those plots, and assemble the data in some kind of a matrix where you can see, okay, plot A has 1% nitrogen, and here we have the reflectance values per wavelength. Then we could use a statistical method such as partial least squares regression that was specifically developed to deal with the high degree of autocorrelation in spectral data to calculate regression coefficients for each wavelength. I'm showing this here. And then we could apply this equation to all the pixel level reflectances within our research area and determine nitrogen content per pixel. The next couple of slides are from a project aimed at mapping vegetation characteristics in the Swiss National Park with APEX, which is an imaging spectrometer. We used the workflow I just showed before to map a bunch of vegetation characteristics, including plant biomass and nitrogen content. And this is at 2 meter squared spatial resolution. Yeah. In this case, we used the resulting maps to investigate the forage resource use patterns of 3 ungulate species that occur at high densities in this area. So for this, we radio-colored individuals and used the GPS data to infer behavioral patterns from the GPS fixes. Then we compared the vegetation characteristics among the species foraging areas. And here you can see, if it runs, yeah, if a time-lapse video of a radio-colored red deer that's moving around and the background is a biomass map. And what you can observe is that this animal really focuses on the green areas. At some point it's going to move over here, yeah, there it is. But it's always the green areas, and those are areas with high biomass content. So using these high-resolution maps of vegetation quantity and quality, we were able to look at forage resource partitioning among 3 species, and we did indeed find that they do separate the forage resources such that the largest species, red deer, which is here, focuses on areas with high biomass, while the smallest species, chamois, which is a sort of small mountain, goat, I'd say, focuses on areas with high nitrogen content. All right, so we know that chemical and structural traits of plants determine their spectral response. Assuming now that plant species differ in their chemical and structural characteristics, we can now start thinking about linking spectra of plants to the tree of life. But before we dive into this, let's take a little step back and think about plant species and their functional and spectral differences. Here you can see the positions of 14 prairie species indicated by different colors in functional trait space. And I'm sorry, there's a lot of information in this little graph here, but what I want you to take away from it is that we cannot find any combination of 2 traits that separate those species. Although we see here, for instance, that grasses, separate already along axis of cellulose content. So they tend to have higher cellulose content and lower non-structural carbohydrate content than legumes and the group of forbs. In spectral space, we see something very different, a lot of overlap. However, when we increase the dimensionality of those spaces and calculate the overlap of species hypervolumes in spectral and trait space with increasing dimensionality, we can see that the fraction of unique hypervolumes of species increase. So unique hypervolumes are spaces that are occupied by one focus species that are not overlapped by any other species. So here in the spectral domain, we selected at random spectral bands, and we found that after selecting at random about 16 bands, we find, almost zero overlap in hypervolumes or 100% uniqueness. In trait space, even if we include all the 10 traits we measured in this study, which include carbon and nitrogen content, pigment composition, and the composition of carbon fractions, we don't reach 100% separation or uniqueness in species hypervolumes. Another way to look at this is in an ordination type of way. And again, you see, some separations of species in trait space, but yeah, not really a clear separation, and more distinct species clusters in spectral space. And what's worth pointing out is that thanks to the high dimensionality of the spectral space, we can, go to, here these are linear discriminant axes that explain not much of the total variation, but that actually do start to separate even the grasses, that tend to cluster together. Those are the blue points. I'm going to show a quick example of species differentiation with spectra, which, based on our previous results I showed you, seems to be possible from spectra. So at Université de Montréal, we were interested in the early detection of the common weed Phragmites australis. And Phragmites is an invasive plant and expands rapidly along the St. Lawrence River system. Using spectral measurements, we trained a statistical model for species differentiation. And again, this model comes from the partial least squares family that was designed for, highly autocorrelated data such as spectra. In this case, it's partial least squares discriminant analysis. And this is a relatively small data set, but still we find high classification accuracies of external validation data. You see the fraction of correctly identified species along the diagonal here. And when we, high classification accuracy also for Phragmites, which is great. And when we confuse Phragmites, actually, we do confuse it with Phalaris. Phalaris arundinacea, which is also an invasive in this area and follows a similar strategy. Perhaps also worth pointing out is that we are, confusing our solidagos quite a bit, and for us, they were also not easy to differentiate in the field. So yeah, that makes sense. So given that we tend to confuse functionally similar and closely related species, we wanted to test if there is a link between species spectral dissimilarity and their functional dissimilarity, as well as their degree of relatedness or shared evolutionary history. We did this by calculating the distances among species in spectral and functional trait space, as well as their phylogenetic distance. And our expectation was that when we plot the pairwise distances of one focal species against all other species, that functional distances among spectrally similar species would be small, whereas when species are very separate from each other in spectral space, they would also be functionally dissimilar. And the same we expected for phylogenetic distances. We expected that close relatives, such as the grasses, if you remember the blue guys over here, close relatives being spectrally similar or have little spectral distance as opposed to distant relatives. And indeed, we found an initial confirmation of this hypothesis looking again at 14 prairie plant species. So yeah, this was a really nice result and indicated that spectral dissimilarity among species increases with functional dissimilarity and evolutionary divergence time. This in part led Dudu to acquire funding for a Nimbus synthesis group, where we compiled probably the largest spectral data set so far. It consisted of over 16,000 spectra from 544 species. And with this data set, Dudu was not only able to spectrally differentiate all major plant lineages across the tree of life, but also he found that major plant clades differ in the regions of the spectrum that are evolutionarily conserved. Is shown here, and that evolutionary rates of spectral regions match evolutionary models of leaf traits, which makes sense and was really a great finding. And this is published in a recent New Phytologist paper. All right, so far we've seen that plant spectral dissimilarity follows their functional dissimilarity, and that species spectral differences are to some degree evolutionarily conserved. We have also seen that spectra differentiate plant species better than sets of commonly measured traits, and this now leads us to this idea of spectra as integrated measures of plant phenotypes. Likely, we can talk about this because spectra of plants capture more of the total variation in chemical, structural, anatomical, and morphological traits than the limited number of functional traits we generally measure. There's only so much we can measure in the field, and yeah, with spectra we just get a very high dimensionality and capture the similarity of plants in an integrative way. So these ideas, were developed to a large part during a Dimensions of Biodiversity project under the lead of Janine Cavender Bears at University of Minnesota with John Gammon and Phil Townsend as co-PIs. And yeah, I encourage you to look up Janine's essay in American Journal of Botany from 2017, which particularly mentions this idea of spectra as integrated measures of plant form and function. Before we dive into this further, why do we care about plant functions and plant functional diversity? One of the early findings from the Cedar Creek biodiversity experiment, where I worked during my time at University of Minnesota, was that above ground productivity increases with functional diversity of the plant communities. This is because plants that are more functionally dissimilar use resources differently, such that together they can use the total available resource pool more completely. Yeah, these results have been replicated several times, and also other positive effects of biodiversity on ecosystem function have been found, including increased stability to disturbance and increased resistance to disease. There are many ways to calculate functional diversity. One way is to standardize traits to common axes and calculate the joint distances among species within communities. Then we can compare communities with each other in terms of their spectral distance or functional distance or functional dissimilarity. But yes, I mentioned before, there is a limit to the number of plant traits we can measure in the field. And sometimes, I mean, it's not that we don't know as functional ecologists what we're doing, but sometimes we might ask ourselves that if the traits that we measure adequately describe all the species characteristics that are important to a specific function. Phylogenetic diversity metrics were developed in part to circumvent this issue of hyperdimensionality of the functional trait space. Phylogenetic diversity is calculated based on branch lengths among species in the phylogeny. The principal idea being that close relatives are more similar with regards to their functional traits and traits in general than distant relatives. An early study by Mark Cadot confirmed the connection between functional and phylogenetic diversity. And this was again done at Cedar Creek. So here we see that that plots with high functional diversity show higher average productivity, and the same is true for plots. The same is true when we look at phylogenetic diversity. Plots that have high phylogenetic diversity are on average more productive. During our Dimensions project, we were able to replicate Mark Cadot's results using our own trait measurements and the newly compiled phylogeny. So both these metrics explain productivity to a similar degree. And we also measured the leaf spectra of the same plants. And we found that leaf spectral diversity explained productivity, again, to a very similar degree than the other two diversity metrics. This is a little bit excessively animated, but the cool thing is, and this is, yeah, I got excited here, I suppose. The really cool thing is that spectral diversity also predicted productivity in this system when it was remotely sensed. So here we used spectral images captured by a headwall camera covering the 400 to 1100 nanometer range at 1 millimeter spatial resolution. And yeah, we found that we can predict productivity from a distance and without determining species identity and abundance, which is pretty cool. As you can imagine, there are now also many different ways we can calculate spectral diversity. Together with Etienne Laliberté and Pierre Legendre at Université de Montréal, we developed an easy to calculate spectral diversity metric that is based on spectral variance. And the nice thing about it is that it fits quite nicely the biological concepts of alpha, beta, and gamma diversity. Since I'm going to talk about alpha, beta, and gamma, I'm just gonna quickly, refresh all of our knowledge, what we mean when we talk about alpha, beta, and gamma diversity. So when we look at this landscape, we can, define a region, if we will, by this yellow box here. Now, within this region, we can find plant communities. Those are the little squares. And alpha diversity is the diversity within one community. So that can be species richness, functional or phylogenetic diversity. Gamma diversity is then the total diversity within the region. And in my case, in the case of the metric I'm going to present, I'm using a beta diversity measure that is non-directional and that is, community dissimilarity. So it is the dissimilarity among community matrices, basically saying how different these communities are from each other. All right, so to develop this approach, we used imaging spectroscopy data acquired by NEON, the National Ecological Observatory Network that is freely available. And here you can see one flight line acquired over Bartlett Forest by ABRIS. Next to it is a false color image of the same region showing the three main axes of spectral variation. We used principal components here just to illustrate, the richness of the data sets in terms of variation that is captured. So this is our regional scale. And we calculate spectral gamma diversity using the sum of squared deviations of each pixel from the average pixel within that region. Sorry the formula, but it's actually not too complicated to do. You calculate the squared distances, sum them up, and then standardize by the number of pixels, and that's all of that. What is interesting is that with variance partitioning, we can calculate the local contributions to spectral gamma diversity, and also what we call the feature contributions. So the contributions of particular wavelengths or spectral components, if you will, to spectral gamma diversity in this case. Because gamma diversity is just one value for the entire region, but the local contribution of individual pixels to gamma diversity can be mapped. It is the sums of squares of one pixel divided by the sums of squares of the total region. And yeah, in addition, what we can do is we can, delineate communities within that region. We chose 40 by 40 meters, which corresponds to a standard size for a forest inventory plot, for example. Spectral alpha diversity is calculated completely analogous to spectral gamma diversity as the squared deviation of each pixel from the average pixel, now per community. Okay, and spectral alpha diversity can be mapped. And dark regions here represent areas of high spectral alpha diversity. We call them hotspots in the paper. But this is not all. We can also look at spectral beta diversity, which we calculate as the squared deviations of each community from the average pixel in the region. And this can also, well, spectral beta diversity is also just one value per region, but the local contributions to beta diversity, so the distances of one community to the regional centroid, spectral centroid, if you will, can be mapped. And again, dark values correspond to high values in this landscape, and by calculating the local contribution to spectral beta diversity, we get to spectrally unique communities, which are communities that likely contain, either rare tree species spectra or rare combinations of more common trees. In the next step, we are using now data from across all NEON sites to investigate if spectral diversity can tell us something about the biodiversity on the ground. And yeah, this was a fun exercise, as you can imagine, working with all these inventory data. And one first step that was quite interesting was that we had to translate different kinds of inventories to a diversity measure as seen from above. So we used The circles are here individual trees that are mapped by neon, and height is recorded and diameter is recorded as well. So we projected them into our plots here. The green is a 40 by 40 meter inventory plot, and we calculated for each plot the crown cover per tree species, taking care of overlapping tree crowns and fun things like that, and then scaled herbaceous plant and shrub inventories to the areas that are not covered by trees. Yeah, I briefly, I think I'm going to skip through the data processing. You can look that up. It's nothing too extraordinary, and I'm sure for many of the steps we're using there are alternatives and perhaps even better ways to do it. We essentially mask pixels that are not vegetation and shaded pixels, and then we performed the PCA and used the main axis of spectral variation to calculate spectral alpha, beta, and gamma diversity. When we look at now spectral diversity plotted against taxonomic diversity per site, we see that some sites, at some sites we find a positive relationship between spectral alpha diversity and the Shannon index in this case. For others, not really. So this is again Bartlett forest, and at Bartlett we find a mix of conifers and broadleaf species. So a combination like this, even if you just have one species of conifer and one broadleaf species, that can result in quite a high spectral diversity, because these are distantly related species and they are spectrally dissimilar. But the Shannon diversity would be low, of course, or the species diversity would be 2 in that example. Another thing we noticed is that we probably need to build a more sophisticated model and take into account differences between different vegetation types. So here the yellow plots are plots from grasslands, green are forest plots, and yeah, this is something to take care of because as you can imagine, we work with 1 meter squared pixels, and it is actually quite a different thing we're getting at. So while with a 1 meter pixel in the forest, we get the spectral reflectance of a tree from one particular individual. In a grassland site, we capture an entire community or a part of a community. So yeah, this is something we have to be aware of also in the interpretation of these metrics. And yeah, just one more example, Santa Rita is a desert site in Arizona with very low vegetation cover. So here also our relationship doesn't look too promising. But we then moved into, the realm of mixed modeling and, modeled the Shannon index in this case as a function of spectral diversity and vegetation type with a side effect. And we found that a high, correlation between spectral diversity and Shannon diversity across all, cover types or vegetation types, if you will. And what's really cool is that the fitted Shannon diversity follows really nicely the measured Shannon diversity across all neon plots. Yeah, and to a final thing I want to show you, and this is a very recent development and I find pretty exciting, is when we think back of this spectral hypervolume thing where plant species separate in spectral space and closely related species clustered together. We thought about other things we can do with spectral hypervolumes, following all inspired by Benjamin Blunder's framework of hypervolumes. And a PhD student, Bjarke Madsen at Aarhus University, looked at changes in spectral hypervolume size of different species in response to nitrogen addition in a biodiversity experiment. And he found that the spectral hypervolumes of species on average declined with nitrogen addition. So here the gray bars, sorry, this is the axes are not really legible, but the x-axis is spectral hypervolume size, and the y-axis is a count. So on average, spectral hypervolume size decreased with nitrogen addition, but it decreased mostly for species that are competitive and slow growing. So the S and C here stands for slow growing, C stands for a highly competitive species following Grimes strategy types. Whereas for faster growing species, the hypervolume size decreased, but it decreased less. Interestingly, for stress tolerant species such as salvia here, the hypervolume size increased a little bit with nitrogen addition, which means that, you know, the plant showed, in response to nitrogen addition, more variation in leaf traits, which, yeah, is interesting to think about. And finally, our C star species, the ultimate competitor in this system, Taraxacum, did not, in this case, you know, the hypervolume size, the spectral hypervolume size did not respond to nitrogen addition at all. This plant seems to be pretty happy where it's at and does not do a thing. Yeah, so in the next step, Bjarke is going to relate the changes in spectral hypervolume size to trait measurements, and this is all pretty new and exciting for us. And finally, I want to make a plug for the Remote Sensing of Biodiversity book, edited by Janine Kevin-DeVeris, John Gammon, and Bill Townsend. It's open access and contains, you know, chapters covering many of the things I talked about, including a great chapter on phylogenetic, you know, a phylogenetic structure within spectra by Dudu, and also nice but more practical chapter on sampling methods by myself. And with this, I thank you so much for joining us today, and I'm happy to answer your questions.
Thank you, Anna. Any questions, everyone?
Thanks, Anna. Maybe I can ask the first question. Thanks very much for your great presentation. I have one question. I just want to know, so for other than, I probably use support for measuring productivity, what type of traits? I know I'm familiar a little bit with nitrogen content, but what other traits do you use to map productivity? And my second question, what portion of electromagnetic spectrum, in particular in the optical region between 300 and 2500 nanometer, are much more important for productivity mapping?
Yeah, to the first part of your question, when we replicated the big bio findings, we measured plant carbon and nitrogen content, the contents of carbon fractions, so non-structural carbohydrates, which are sugars and starches, cellulose, hemicellulose, and lignin, as well as pigment composition, so chlorophyll, the contents of chlorophylls, carotenoids, and xanthophyll cycle pigments. Yeah, this is what we used. And regarding the spectral regions most important for biomass prediction, you know, I I would have to go and look at the loadings of a PLSR model, but for predicting productivity, we can also approximate this by a simple greenness index, such as the NDVI, which uses a wavelength in the red and in the near infrared. So it is, the degree of absorption of photosynthetically active radiation normalized by reflectance in the near infrared, which is not affected by photosynthetically active radiation in any way. So I would for sure say, well, it is a difficult question, right? Because we know that the pigments are important for photosynthesis.
Yes, no, my question is because most of the time when we have access to hyperspectral data in 400 nanometer to 1000, I just wanted to know the role of short infrared portion for productivity mapping. So if we only have access to that region, it still can be map productivity.
Yeah. So I'm sure that wavelength regions in the near and shortwave infrared also have an influence, because pigments are not the entire story. It's also a matter of how dense the vegetation grows, how high it grows. So it's a matter of canopy density, canopy complexity, leaf area index, and so forth. And these traits influence more the near and shortwave infrared.
Perfect, thanks very much. Because you definitely, as you mentioned, like measuring chlorophyll content, like pigment contents, leaf area index, we can just, so we can use that 400 to 1000 region. But I just want to know if even we can estimate nitrogen content. So with only using that region, not incorporating short infrared bands.
So, yeah, it's a good question. I mean, what I do when I'm specifically interested in proteins, as opposed to chlorophyll, is that I actually cut the visible part of the spectrum out, because the visible is clearly influenced by pigments, whereas proteins, which are larger molecules, are more influencing the longer wavelength regions. So I'm for proteins, I'm specifically moving to the longer wavelengths, if that makes sense. Yeah.
Thanks very much. By the way, the picture you showed took me to 10 years ago, because I met Dr. Austin when I was a PhD student. So in Japan. So, and I had I spent 2 days with her in Japan. And also you mentioned Dr. Gammon. It was just 2 weeks ago. I had 2 of my graduate students here today. So we talked about, we were talking about PRI, photochemical refractive index. So I used some non-imaging spectrometer data to calculate PRI and correlate it with several narrowband vegetation indices. So anyhow, so it took me to a long time ago. Thanks very much for your great presentation.
Thank you.
Anna, I have a question. With regard to using hyperspectral imagery for operationally mapping species that have similar spectral characteristics, where do you see the state of science in that regard? Where it's really challenging with sort of standard multispectral imagery such as Sentinel and Landsat to really discriminate species that are spectrally quite similar. So do you see, I mean, sort of think of it in an operational context. Is this something that we can really leverage, hyperspectral tools, hyperspectral imagery to really map species at a higher degree of specificity at a landscape scale in an operational context?
Yeah, I think, in terms of where we are at, we are in the development and testing phase with this. I think, the NEON data set is going to be like really interesting for investigating where we can get at, because, for species mapping in a forest, we need high resolution data in general. So when we work with, like NEON provides 1 meter squared resolution data. And the trick for training a species identification model, I would say maybe there's going to be a two-step process to it. So first, field data is really essential. So with these statistical tools we're using, they're not working if we don't have good field data. So we need data from the ground that identify trees and say, okay, this is species A, this is species B. Data that are spatially accurate, and are georeferenced. And then we can match pixels from the imagery to those trees and train a model. In this regard, I think that the second step that can make the method operational is to leverage also species inventories. So, in general, we do have a relatively good sense which species we find where. And when we limit the problem to fewer species, algorithms do better, of course.
Right.
Yeah, it's not operational yet, but I'm very optimistic. Especially, also, with the satellite missions that are coming, that are gonna capture the data multi-temporally. And in deciduous, I'm just looking at your background, Tony, in our deciduous forest systems on the East Coast. I mean, it's going to be really nice to also leverage fall data, for instance, to differentiate trees. And that should make the problem a bit easier.
Right. Yeah, it's a tough problem for sure. Great, thank you. Any other questions, anyone?
A quick one along the same lines of Tony's in terms of like the, how to make hyperspectral imaging, like how to make that work for like fine scale mapping. Would a spectral library that is generic, say leaf level stuff, does it help? Does it work that I have like a sugar maple measured, at the leaf level measured in Maine to predict how it's doing in like somewhere in Ontario? Or, how like when you show those models that have like a site effect, like how important is that? Can you clump those sites into a more generic thing? Because at some point it kind of like defeats the purpose, right? I mean, if you have to go in and measure leaf level things everywhere, you might as well just do the inventory.
Yeah.
Right? So how, I'm being kind of a
Sure.
advocate here, but do you get that sense of like how how much field data from the like a close by location do we really need to get this going?
That's a great question. So from my experience, I don't think leaf level spectral libraries are going to help us currently all that much with species mapping from remote sensing. I tried it with the high resolution headwall data at Cedar Creek, using leaf level data, train a model, apply it to the headwall, which is, super close to the ground. I only picked the best pixels that are super sunlit and nice, but yeah, it didn't work in my case. Maybe someone will figure out something really smart, but I don't have very high hopes at this current point. But what does work well is when you go into the image data and select pixels from crowns. So if you want to predict species distribution from airborne imagery, let's say, you should train your model with spectra from airborne imagery. And to your point of differences, like regional or local differences in species spectra and their traits and how this is going to influence model performance. Well, I do think that local models generally outperform larger scale models. However, Phil Townsend is working on, the trade models for the neon sites, and his models do actually a really nice job predicting trades all across sites. And Phil, when I remember correctly, in essence says the same thing. It is possible to make the model even better if you focus all your attention on one side, but you can still get a decent model with, important is of course to know uncertainties and things like that, but it is possible to get a decent model for traits that works across all ecosystems that are covered by neon in that case. And I think, since PLSR for trait prediction and PLSDA, they come from the same family, they work pretty similarly. I would expect that this is similar for species predictions.
Mm-hmm.
Of course, if you want to map, say, Quercus alba across the entire United States, it would be good if the model includes Quercus alba spectra from across all of the United States, because you do have to incorporate all that variation in your model in order for it to work well.
Great, thank you. Any other questions, everyone? All right, well, thanks again, Anna. Thank you very much. This was really wonderful. And thanks everyone for joining us. Have a great afternoon.
Everyone. Thanks for the questions. And yeah, it was my pleasure.
Great.
Take care, everybody.
Thanks again, everyone.
Bye-bye.
Bye.